In this lecture video, I will discuss the general characteristics of organisms we classify as prokaryotes. So we will not delve into the nitty-gritty details. Instead, we'll focus on their general characteristics, leaving the details to your microbiology course. So prokaryotes are found almost anywhere on Earth. Okay, in the most extreme of places, visually there might seem to be no life in existence, but it's actually very possible to have prokaryotes living on the surfaces of those places. So even in your stomach, where the acidity is very, very low, okay, or the pH is very, very low, prokaryotes can still survive. So other places that are too extreme for the usual type of life forms may be very salty places, like the waters in the Dead Sea, or very cold places that are permanently frozen, just like the ones under the ice sheets in Antarctica, or very hot places, like in hot springs near volcanoes. So prokaryotes may be divided into two domains, or they are divided into two domains: domain bacteria and archaea. So generally, the cells of these organisms are a lot smaller in comparison to the cells of eukaryotic cells. Okay, so generally the sizes are between 0.5 to 5 micrometers, okay, as written here, as compared to 10 to 100 micrometers of cell size for eukaryotic cells. And the cells of these prokaryotes may be roughly classified into three shapes. Okay, the spherical bacteria or spherical prokaryotes, we call them as coccus or cocci. Okay, and then we have the rod-shaped prokaryotes, the bacilli, and then the spiral-shaped prokaryotic cells. So here are representative microorganisms or prokaryotes that have cells as described. And they are so small that in this particular electron micrograph of the tip of a pin, okay, they can actually crowd, like thousands of them can crowd at the tip of the pin. Okay, so this is the tip of the pin, and you can see here hundreds or maybe even thousands of rod-shaped bacteria. So this is the general characteristic of the cell of a prokaryote. Okay, so prokaryote, first and foremost, doesn't have a nucleus to store its DNA in, and it has a very simple circular chromosome instead of the chromosomes that we have, you know, as humans or even other eukaryotic cells. They also lack compartmentalization in their cells, like there are no Golgi apparatuses, there are no endoplasmic reticula. Instead, they have just the cytosol, okay, and floating around in the cytosol are protein synthesizers, okay, which are known as the ribosomes, okay, as you might have observed here, okay, the brown dots, okay, seen there are ribosomes. Nevertheless, the cells can move, okay, many of the prokaryotic cells can move with the virtue of flagella, as we can see here. Okay, so the flagella of these prokaryotes are very different from the flagellar structure of eukaryotes, as we will see later on. And here you have structures that seem to look like cilia, but they're not cilia. They're known as fimbriae, and they are used by the prokaryotic cell to attach onto surfaces. And then here we have the pilus. Okay, the pilus is used to connect with another prokaryotic cell in the process of conjugation to exchange with them genetic materials. So, prokaryotic cells, just like eukaryotic cells, have cell membranes, okay, as illustrated here. Okay, these are cell membranes. And prokaryotic cells can be divided into two, most specifically bacteria, in terms of the structure of their cell membranes and the cell wall. Okay? The cell wall of bacteria are known as peptidoglycans, and they are made of connections of carbohydrates and amino acids. Okay? And you may classify bacteria into two depending on how their cell walls and cell membranes are arranged. Bacteria which have a single cell membrane underneath a thick cell wall, like the one here, okay, are known as gram-positive bacteria. Okay, so this naming is due to their reaction to a particular set of stains whenever they are stained. Okay, the gram-positive bacteria will retain the purple stain that's done during gram staining. So as you can see in the picture here, the gram-positive bacteria are the purple ones, the cocci that you see here, the purple cocci. Okay? The gram-negative bacteria have thinner cell wall. Okay. So the peptidoglycan layer is thinner. Sorry, the cell wall is actually quite thick. I was referring to the peptidoglycan layer, which is thin, because it's sandwiched in between two cell membranes, an inner one and an outer one. Okay? And this particular arrangement of the membranes in the cell wall causes the gram-negative bacteria to not retain the purple stain that's placed during gram staining. As a result, they appear a lot paler than the gram-positive bacteria after gram staining. Here, they are the pink-colored bacilli. So the gram-negative bacteria are also more resistant to antibiotics because many antibiotics work by destroying this peptidoglycan layer so that water can rush into the cell and lyse the cell. But for gram-negative bacteria, because of this outer cell membrane, okay, some antibiotics have difficulty in penetrating to the peptidoglycan layer. Prokaryotes have also adapted, many of them have adapted to living in really harsh conditions. And some of them to the point of undergoing what we can almost say as hibernation when the environmental stress is very, very high and it's unfavorable to live. So these bacteria can actually form what we call as endospores, which can remain alive even when the environment is very, very extreme already. So some of these endospores can even survive heating. Okay? So this is how they can actually survive through very harsh environmental conditions. So we can see here a bacterium that has started to form an endospore inside of it. Okay? In time, this bacterium that I've outlined here will die because of environmental stress. But it has sort of transferred its life into this endospore that's forming inside of it. So when the bacterium dies, the endospore here continues to live on. And when the right environmental conditions come again, this endospore can become an active bacterium again. Okay? So, as mentioned a while ago, they have membrane that surround their cells that help them attach onto surfaces. Okay, as shown here. So, they look like cilia, but they're not cilia. Okay? And there are specialized longer projections we call pili or pilus, okay, that can connect to other bacteria to exchange genetic material. So here we have an illustration of the microstructure of the flagellum of bacterium or bacteria. Okay? Unlike the flagella of eukaryotes, which are powered by motor proteins, okay? You remember your motor proteins? The flagellum of bacteria are mainly powered by the base here, okay? Which acts like a generator motor that can turn, this can actually rotate, okay? And as this rotates, the bacterium itself is propelled forward or backward by the rotating flagellum. Okay, so here we have an electron micrograph of how that motor of the flagellum actually looks like. Although the internal structures of bacteria or prokaryotes at that are much simpler than eukaryotes, we can see here how folds of the cell membrane can actually start forming inside of the cells of these prokaryotes. Okay? In fact, here in this photosynthetic prokaryote, you can see very complex foldings of photosynthetic membranes that contain chlorophyll, much similar in appearance to your endoplasmic reticulum in eukaryotes. So because prokaryotes don't have nuclei, it's very easy to extract their DNA. Okay? So here we can see the DNA or the whole chromosome of a bacterium. So this is the bacterium. And the whole chromosome is coming out of the bacterium. Okay? And this is actually circular. So if you can unfold these, okay, you will find that this one chromosome is circular. And then there are smaller portions or there are smaller DNAs that float in the cytoplasm of the prokaryote here that separate from the chromosome, and we call them the plasmids. So here are some plasmids that have come out, and many plasmids are actually responsible for giving the bacterium or a group of bacteria a characteristic that can withstand antibiotics, so antibiotic resistance. So prokaryotes, because they lack nuclei, and they lack the machinery for proper mitosis, okay, or meiosis, they can divide themselves or reproduce by dividing their cells using a process called binary fission. Okay? And binary fission takes a lot shorter a time than, let's say, mitosis. So let's say if we start with one bacterium at the beginning of the day, after 24 hours, that one bacterium undergoing binary fission can become millions of bacteria already, which are sort of clones of that bacterium. Okay? So, because bacteria or prokaryotes in general are very fast in reproduction, their rate of mutation, as I mentioned, is also really, really fast. So it's very easy for them to adapt to their environment. And it's also very difficult for us to target all of them with a specific antibiotic because at a specific time frame, some of them may have already mutated to become antibiotic resistant. Okay, because they're very easy to mutate. And they can even share their genetic material with other prokaryotic cells, and in a way, they can share their antibiotic resistance to other bacteria or prokaryotic cells. So here we have an illustration of how a virus can actually incorporate its genetic material with that of the bacterium. Okay? And in this process is called transduction, and this is one way for a prokaryotic cell such as this bacterium to actually mutate. So conjugation is like the sexual reproduction between prokaryotic cells, okay, but not really. Because conjugation is when they transfer genetic material from one cell, the donor, to another cell, the recipient, using, of course, the structure we call the pili. Okay, so here's an example. So one bacterium here might be transferring its genetic material for antibiotic resistance to another bacterium. Thus, both of them will become antibiotic resistant after this conjugation process. Okay, and here are other possibilities of conjugation. It can either be the plasmid that's going to be transferred from one bacterium to the other, okay, as shown here in letter A, or it could be a whole portion of the chromosome of the bacterium that can be transferred to another bacterium, as shown in letter B. Okay, so these antibiotic-resistant plasmids, of course, will render the bacterium antibiotic resistant. Now, here we have different modes, nutritional and metabolic adaptations of prokaryotes. Okay, so when we hear the word phototroph, okay, that means they can obtain their energy from light. Okay, then we have here chemotrophs, meaning to say they can obtain their energy from chemicals. Now, that's in terms of energy. Okay? But living things don't just need this energy. They also need building materials for their cells. And that's no other than in the form of organic materials with carbon in them. And then we can classify organisms into two in terms of how they get their carbon. They can either be autotrophs, okay? They require inorganic sources of carbon to build themselves. And then we have heterotrophs, which get their building blocks for themselves by eating organic materials, such as how we do it, humans. We eat organic materials. So therefore, you can classify prokaryotes and living things in general according to how they get their energy and how they get their building materials. Photoautotrophs, for example, get their energy from light, and they use inorganic sources of carbon to build themselves up. Chemoautotrophs get their source of energy or get their energy from chemical reactions, and they get their building materials for themselves from inorganic carbon sources. Photoheterotrophs use light, okay, as a source of energy, and they get their building materials for themselves from organic sources, meaning to say they eat other organic sources or living things. And chemoheterotrophs, like us humans, we get our energy from chemical reactions. and we eat organic materials to build ourselves. So for prokaryotes, this is very important for them. Okay? Many prokaryotes are obligate aerobes. Okay? Meaning to say they really require oxygen. Okay? For cellular respiration. Okay? That's how they harvest their energy from their food. And humans, even if we are not prokaryotes, okay, we belong to this group, obligate aerobes. Okay? We really need oxygen in order to do cellular respiration. Then there are obligate anaerobes. Okay? When the earth was young, it was hypothesized that there was no oxygen at all in the atmosphere of the young earth, and all of the living things that once were did not need oxygen in order to survive. In fact, when oxygen came, when cyanobacteria started producing waste products of oxygen, those obligate anaerobes were poisoned. They are poisoned by oxygen. So presently, we can find these obligate anaerobes in areas where oxygen is difficult to come by. Like, for example, underneath the ocean, okay, near hydrothermal vents, where free oxygen in the form of gas is very difficult to come by. And then we have facultative anaerobes. Okay, these are the microorganisms or the organisms that may live, okay, with oxygen, and they can also survive without oxygen. An example of this will be your E. coli, the one found in your colon, which can survive without oxygen. That's the reason why they can survive in your large intestine, even if the amount of oxygen there is very low, because they are facultative anaerobes. And if ever you poop them out, they can still survive because the oxygen in the atmosphere will not kill them, because again, they are facultative anaerobes. Okay, so nitrogen is also a very important nutrient, okay, in living things. And for prokaryotes specifically, they have evolved certain adaptations to fix atmospheric nitrogen into something useful, like ammonia, and even into nitrates. Okay? And we call this fixing of nitrogen, nitrogen fixation. And there are organisms that actually, I'm talking about prokaryotes that actually specialize on this. Okay? Number one, you have your mutualistic rhizobium bacteria in the roots of leguminous plants that can produce their own nitrates. Number two, we have the cyanobacteria that may look like they are multicellular organisms, but they are really just a colony of cells that have come together so that they can help each other survive. Now, in that colony of cells, as we see here, these are not multicellular organisms, but these are individual cells that we see here. They can survive by themselves. They have grouped themselves together because some cells, known as heterocysts, like these two, can actually use atmospheric nitrogen and produce nitrates or ammonia out of the atmospheric nitrogen, which then these photosynthetic cells can use because they need nitrates as nutrients. And in return, the photosynthetic cells produce food that the heterocyst cells use as food as well. Okay? So it's like a colony where there's a mutualistic relationship between these two types of cells. Okay? And some bacteria and many prokaryotes have actually evolved because they are unicellular organisms. How can they survive in long terms? They have actually evolved in a way that they can interact with other prokaryotes, and they can stick together and form what we call as biofilms. Okay? And in these biofilms, they act like as if they are a colony or a multicellular organism. And these biofilms can be seen here. Okay, here are some examples. Inside water pipes that have never been cleaned, bacteria may accumulate and form these slimy structures, and these are actually biofilms. Okay? If you haven't brushed your teeth in a while and you try to scrub them with your fingers, you might feel that they are a bit slimy. That's because bacteria or prokaryotes have already accumulated on them in the form of biofilms as well. And if you leave them there for a very, very long time, they can form plaques, as shown here. Okay? Which can then, when they are together, their waste products, which are acidic, can actually erode away the enamel of your teeth. Okay? That's why you will have cavities. And then they live through eating the remaining foods that you eat, especially sugar. They love sugar. So if you're a sweet tooth and you hardly brush your teeth, bacteria on your teeth will form biofilms really, really fast. And here, this is the opening picture from the chapter in your book. We can see biofilms, this time, of archaea forming underneath this salty water of the lake. So these archaea here are attached at the bottom of the lake bed, okay, where the waters are salty. So in the next part of this presentation, we are going to see an overview of the different types of prokaryotes, especially bacteria.